The degeneration and regeneration of nerves. Nerves can degenerate, nerves can regenerate. Okay, regeneration can occur and if certain substances or chemicals are present or are provided. That's when we shall have regeneration, and we shall talk more about it in this very video. If it's 7 Science Theater Uganda, don't forget to subscribe for more and better videos. Okay, don't forget to subscribe for more and better videos. Like our videos, share to other people to also enjoy. Right, let's start right away. A neuron can degenerate. In case you injure the axon of a nerve, okay, what will happen? There will be a series of degenerative changes which will be seen at three levels. One, we can see these changes listed to the injury, okay, within the axon. We can see it within the axon but proximal to the site of injury. And still we can see the changes of degeneration within the cell body, call it the soma. So these three occurrences are at different sites of the neuron with regards to the site of injury, okay? And what are the common causes of nerve injury? One, we can have transition through and a through cut and cause degeneration. Two, if you crush the nerve fibers, okay, they can get injured. Three, local injection of toxic substances. Sorry for this. A local injection of toxic substances is another cause. Four, we can have ischemia due to obstruction of blood vessels. Hypoperfusion and associated ischemia will cause injury of the nerves. And now we have other effects of hyperpyrexia of the neurons. These are causes also of nerve injury, hyperpyrexia, okay? And if we continue, how is the nerve injury graded? We can grade nerve injury based on this man Sunderland, who graded them in terms of degrees. For example, he made 5 degrees. Like nerve injury can occur in 5 degrees, and the first degree is that it involves is the first degree, which involves only transient loss of function resulting from mild pressure on the nerve, and the loss of the nerve fibers returns within few hours. Loss of dysfunction. Here there is no anatomical damage, but just a mild pressure on the nerve. Loss of function occurs, and this returns within a few hours to few weeks of the removal of the causative pressure, since the axon is not destroyed. So if you remove the pressure, then the function will be regained. Second degree injury occurs and includes severe nerve damage with intact tendoneural tube. It results from severe and prolonged pressure on the nerve. The regeneration and restoration of the function of the nerve are facilitated as the endoneural tube remains intact. Because the endoneural tube has remained intact, then regeneration and restoration of the function of the nerve will be facilitated. Okay, so here there is damage, severe nerve damage, but the endoneural tube is intact. The third degree is that it will include severe damage to the nerve fiber, but here the endoneural tube will be interrupted. And the fourth degree involves severe damage to the nerve fiber, which is associated with the disorganizing of the nerve fasciculi. In the recent video, a video where I talked about a neuron, I showed you how the peripheral nerves are organized from a single axon covered by several layers to form fascicles or fasciculi. So these are here the nerve fibers will be disorganized now in the fourth degree, okay? And the fifth degree injury is labeled when there is or when there occurs complete transaction, i.e. the nerve fibers are cut through and through, okay? So here at 50 degree is very bad to an extent that just immediate degree injury. You'll find that just after injury, there develops degeneration, starts immediately, unlike these other forms of degrees of injury, according to Sunderland. So let's continue. And the changes in the cell body of a neuron. We said we have changes in the axons, and we have changes in the cell body. So in the cell body, these changes will start just after 2 days, up 15 to 20 days. And the changes include: one, the new cell substances will undergo chromatolysis. Talked about this recently, that new cell substances undergo disintegration and dissolution. Okay, and the Golgi apparatus, mitochondria, and neurofibrils are fragmented and they eventually disappear. All this is occurring during degeneration. And the third change is that if you look at this, the cell body, I can summarize it here for you, some few, then we shall continue with other changes. Here in the normal, we have nuclear bodies very intact, nucleus, okay? But now, the nuclear bodies are disintegrating, chromatolysis, okay? They are starting to disappear. Now the mitochondria break down and also disappear. The Golgi, now the nucleus goes to the periphery, as you can see. It is displaced from the center to one pole of the soma, okay? And yeah, basically that we have a lot to talk about. Let's continue. That the cell body draws in more fluid, enlarges and becomes spherical, unlike how it was before. And the nucleus is displaced to the periphery towards the cell membrane. And sometimes the nucleus is extruded out of the cell, in which case the neuron atrophies and finally disappears completely if the nucleus is extruded out of the cell. And if you continue, if you look at this, there are still more changes, but I want to show you just some few things here. There's degeneration. You can see the axon. This is the site of injury, and the changes are occurring distal. Degeneration can occur distal, proximal to the site of injury, and within the cell body, which I didn't show here. And now regeneration will start. As you can see, you can take a note of this structure here, which is dividing into a couple of them, and one is entering inside, and now it is becoming an axon, which is myelinated after 1 year, probably, yeah, 1 year. So there's a lot of changes which occur, okay? So let's continue. And another change is that the axis cylinder becomes swollen and irregular in shape within a few hours of injury. And after a few days, it breaks down into small fragments, the neurofibrils, into small fragments. Now the neurofibrils within it will break down into granular debris and seen in the space occupied by the axis cylinder. So we shall see granular debris appearing now, and the myelin sheath will show slow disintegration. In fact, myelin sheath is converted into fat droplets containing cholesterol. It shows slow disintegration. In fact, it will be converted into fat droplets which contain cholesterol esters. And the neurolemma sheath is usually unaffected, but the swan cells start to multiply rapidly. We shall see the significance of this rapid multiplication during regeneration. To the macrophages, these macrophages in the body will invade the region and remove degenerating axons, myelin sheath, and also remove cellular debris, and thus neurolemma tube becomes empty. So when the neurolemma tube becomes empty, now the swan cells cytoplasm proliferates rapidly and fills up the empty neurolemma tube. These cells produce a large series of membranes that help to form numerous tubes, which play a vital role in the regeneration of nerve fibers. So let's continue. Now we've gone to the regeneration changes. Regenerative changes are very many, but not too many. Let's continue. We have anatomical regeneration and functional regeneration. But in anatomical regeneration, there are changes which occur within the axon. First, there is a stage of fiber formation, where the axis cylinder from the proximal cut end of the axon elongates and gives out fibrils in all directions. These branches grow into the connective tissue at the site of injury in an effort to reach the distal cut end of the nerve fiber. Okay? And there is a stage of entry of fibrils into the neural tube and the neural tube. So first we formed the fibrils from an axis cylinder. Now we are saying we are having the entry of each fibril or fibrils into the endoneural tube. The strands of axon cells from the distal cut end of the axon will guide the regenerating fibrils to enter the axon endoneural tube. Once the fibril enters the endoneural tube, it grows rapidly within it. The axon fibrils that fail to enter one of the tubes degenerate. So, if a fibril fails to enter one of the tubes, it will degenerate. And if two enter, only the largest one will survive, and the smallest will degenerate. Okay, so there is a stage of active growth. This stage, axon fibril grows through the endoneural tube, enlarges and establishes contact with an appropriate peripheral end organ. The new axon formed in this way is devoid of the myelin sheath. Therefore, myelin sheath will be formed and attached or will be deposited by the Schwann cells only, and this myelination is completed after around a few years. So what are the changes in the cell body of the neuron during regeneration? These were the recent ones were changes in the axon during regeneration. Now what are the changes in the axon when there is a regeneration? We're going now to the changes in the cell body when there is a regeneration. First, nestle granules followed by Golgi apparatus will appear in the cell body. Because nestle granules are representing protein synthesis, so they will be needed, they will appear first. We need proteins that synthesize enzymes and other material which will be important in regeneration. Now the cell loses excess fluid and regains its normal size. That is occurring in the soma. The nucleus occupies now a central position, and the above changes in some of the neurons start within 20 days of injury, and by 80 days they will be completed. Okay, now we've turned to functional regeneration. The previous has been anatomical. The functional regeneration, all along we've been describing anatomical regeneration. So the functional regeneration will occur, but it will take a lot of time. It will occur after a long period of time. Let's continue to the factors that promote neuronal growth. Okay, these are called the neurotrophins, and the neurotrophins are proteins which provide trophic support to the neurons. They promote nerve growth and survival. They also have a role in the neuronal plasticity. We talked about this characteristic or property of neurons the within the recent video about synaptic transmissions. Okay, and the neurotrophins are produced by the muscles basically and other structures like the glands where the neurons innervate. Okay, so those are the ones which produce the neurotrophins, and maybe some can be produced by the astrocytes. Okay, and the established neurotrophins we know include nerve growth factor NGF, brain-derived neurotrophic factor, and neurotrophin 3, 4, or 5. Okay, these are the neurotrophins which we know basically: nerve growth factor, brain-derived neurotrophic factor, and neurotrophin 3, 4, and 5. Okay, other growth promoting factors, we have the ciliary neurotrophic factor from the eyes, have leukemia inhibitory factor, we have the insulin-like growth factor, that is somatomedin C. We have transforming growth factor, we have fibroblast growth factor, we have platelet-derived growth factor. All these are also neuro growth promoting factors. Okay, so that is all. Don't forget to like our videos, subscribe for more and better videos, like our videos for more notifications, and share to other students